In the previous videos, we've talked about both the bacteria and the archaeal domains. Now let's take a few minutes and look at the eukarya domain. We'll start by looking at the fungi. Fungi are very interesting organisms, and we could spend an entire term discussing just this one particular group. But we don't have that opportunity in a course like this. It's important for us to know, though, where the fungi fit in terms of microbiology. There are some fungi that are microbes because they're single-celled and microscopic. But fungi can also be multicellular, and in some life stages, they are macroscopic. When we compare them to the bacteria and the archaea, fungal cells do have a cell wall outside of their plasma membrane. But in the case of fungi, that wall is made out of a polysaccharide carbohydrate called chitin. Like peptidoglycan, chitin is a relatively strong material and is protective for the fungal cells that it surrounds. In fact, chitin is also the type of carbohydrate that composes the exoskeletons of insects. If you can remember the last time you stepped on a bug on the sidewalk and you heard that characteristic crunching sound, that crunch comes from the chitin in the exoskeleton. So it's not a true cell wall like the peptidoglycan cell wall around bacteria. It's different from the S layer around archaea. It's its own version of a protective outer coating made out of chitin. When we compare the fungi to bacteria and archaea, they have a relatively broad array of reproductive processes. Unlike the bacteria and the archaea, which use asexual methods, fungal cells can reproduce both asexually and sexually using several different processes. Also, in contrast to the bacteria and archaea, the fungi use a relatively limited array of energy sources. Remember, we said that both bacteria and archaea can use organic sources, inorganic sources, and even for some species, sunlight as their food or their source of energy. The fungi are different. Fungi rely on organic materials as their food source, as their energy source. Now, when we look at the diverse array of fungal organisms, we can put them into one of three general categories. Number one are the yeasts. Number two are the molds. And number three are mushrooms. Most of us are very familiar with mushrooms. Mushrooms are actually reproductive structures for fungi. They are multicellular, and of course they're macroscopic. We can see them with our eyes. These are what are referred to as the fruiting bodies of certain species of fungi. And of course, while we do eat many different kinds of mushrooms, there's a variety of other mushrooms that are quite toxic to us as humans. Molds are very different. Molds grow in the form of what are called hyphae. And hyphae are simply multicellular filamentous structures, long linear structures that branch into what we refer to as filaments. You've seen mold growing before. You've probably seen it growing outside of your home, as well as perhaps in places like your bathroom, in your shower, or perhaps in your kitchen. Yeasts, the last category for fungal cells, are single-celled structures, and yeasts can be found again in various environments, both inside and outside of our homes. On this slide, we're looking at the three forms of fungi. We're looking at, on the upper left, molds that are growing on a petri dish on an agar surface. And you can see there's a variety of different species of molds growing here. On the right-hand side, this is actually an electron microscope image of yeast. So the thing that I like about this particular image is that not only does it show us the sort of ovoid shaped yeast cells, but it also shows that many of these cells are reproducing, and they're using an asexual form of reproduction called budding. You can see that there are parent cells like this one right here, and then you can see these little projections, these little circular projections sort of growing off the surface of the parent cell. Those are the buds. Those are daughter cells, and they will grow to a certain size, and then they'll be released from the parent cell. Down at the bottom here, this is an image that shows a wide variety of edible types of mushrooms. Only some of the many types of mushrooms that humans like to eat. Next, we're going to move over to the protists. Now, compared to the fungi, the protists are unique in that they are not one group of organisms. When we think about the protists in terms of being one of the types of eukaryotes, protists are sort of the extra group. When early scientists in history were categorizing eukaryotic organisms, they recognized animals and plants and fungi, but they also recognized that there were many other types of organisms that just didn't fit into those three categories. And what made these other types of organisms problematic was that they weren't all really closely related to each other. In other words, from an evolutionary standpoint, these were individual organisms that were sort of scattered all over the tree of life. So, a group was developed called the protists. So you can think of protists as being organisms that are eukaryotes, but that don't belong to the animal group, the plant group, or the fungus group. Now, when we're in a microbiology course like this one, we tend to think about protists as including the algae, and that's what we're looking at on this slide, as well as what are called protozoans, and finally slime molds. The algae and the protozoans in particular fall into our definition for microbes. So let's begin with the algae. Algae are single-celled or multi-celled organisms. They can be microscopic or macroscopic. Now, obviously, when it comes to microbial algae, we're talking about the single-celled microscopic varieties. When we look at these cells under the microscope, we can see that they're surrounded by, once again, a type of cell wall. Unlike the other types of cell walls that we've discussed so far, the wall that surrounds an algal cell varies. It can be made out of any of several different types of materials. So it totally depends upon the genus and species of algae that you're examining as to what material its cell wall will be made out of. For example, the agar that we use for culturing microbes in the laboratory comes from algal cells, from certain species of algae, and it makes up the wall that is outside of the plasma membrane. There are other types of algae that have a wall made out of a a material called carrageenan. There are actually, again, several different types of materials that make up the cell walls in algae. Algae are capable of using both asexual and sexual methods for reproduction, and like the fungi, they have a very narrow source of foods for energy. They are photosynthetic organisms, so they are dependent upon the process of photosynthesis to harvest energy from the sun as a source of energy. On this slide, there are all kinds of drawings of different types of algae. If you've ever been to the seashore, if you've ever sort of wandered into growths of seaweed, you've seen algae. If you've ever gone to one of New Hampshire's beautiful lakes in the summer, you may have seen algae growing. Algae has a tremendous variety of life forms. Remember, some of it grows as single-celled microscopic organisms. So those you would only be visible to us underneath a microscope. But there are an incredible range of macroscopic forms of algae. And again, these you would see most frequently as seaweed at the shore. Now let's move on and think about the protozoa. If you take that word protozoa and you define it, if you translate it, it translates into early animal, protozoa. So one of the most notable traits of the protozoa is their animal-like quality of movement. Protozoa are always single-celled and microscopic, so they fall into our definition for microbes. They can be free-living organisms, or some species are parasitic. And by parasitic, what we are describing is the relationship that these organisms have. They are dependent upon a host, and while they benefit from the relationship they have with the host does not benefit. The host is actually harmed by that relationship. That's what a parasite is. It sort of takes advantage of a host organism. It benefits by living in conjunction with that host organism. But the host organism is harmed by the relationship. So yes, there are parasitic protozoa that cause diseases in humans and other organisms. Something else that's very unique about the protozoa is that they live through very complex varieties of life stages. And what I mean by that is that they have what we refer to as a proliferative stage or a growing stage. And they also have what's referred to as a dormant stage, or what's often referred to as a cyst stage. So throughout both of these, the proliferative and the dormant stages, there are several steps to these cycles of life that can become very complex depending on the species that you're examining. Unlike the other organisms that we've talked about so far, the protozoa are unique in that they do not have any kind of external wall structure outside of their plasma membrane. So just like the cells inside your body, the protozoa has a plasma membrane with nothing else outside of it. They're capable of using both asexual and sexual methods of reproduction, and they have a relatively limited source of molecules that they can use for energy. Specifically, they rely on organic molecules. Now I have a list of some of the more common pathogenic protozoa. These include things like certain species of amoeba, what are called Giardia, Toxoplasma, and Trichomonas. If you look at the pictures on the right-hand side of the slide, up at the top you can see an image taken with a microscope, or an ordinary light microscope, of an amoeba. Now there are lots of amoeba out in the world that are perfectly harmless to us, but there are a few species of amoeba that are pathogenic. The image right below the amoeba, this is a Giardia. Giardia are most commonly associated with freshwater sources and undercooked foods, particularly undercooked meats. You see Giardia causing disease in humans, oftentimes when humans take water out of a stream or a river and drink it without purifying it first. Giardia infection causes a gastrointestinal type of illness in humans. The next image, again taken with a microscope, this is the Toxoplasma organism. This happens to be Toxoplasma gondii, and this organism is often found again in undercooked meats. It can be found in cat feces. It can be transmitted from a mother to her fetus across the placenta. Toxoplasmosis is an illness that creates flu-like symptoms in the people that get it. But it can become quite problematic. It can lead to things like lung problems. It can lead to damage in the eyes. It can produce seizures. In the people who are particularly susceptible to it, toxoplasmosis can be a very serious disease. And lastly, on the bottom here, we have an image of an organism called Trichomonas. Trichomonas causes one of the sexually transmitted diseases called Trichomoniasis. This is a very common sexually transmitted disease in our country. As you can see, the organism has a somewhat similar shape to the Giardia organism. It has sort of this teardrop shape. You see that in both of them. And of course, because they're both protozoa, these are also mobile organisms. They're capable of swimming, they have movement. The scary thing about trichomoniasis is that about 70% of the people who get infected with it have absolutely no clinical signs. People that do show signs, and primarily we see signs in women, tend to report typical sort of itching, burning around the genitalia. Sometimes there's a discharge associated with trichomoniasis. So we see clinical signs more often in women than men, but it is a curable sexually transmitted disease. So on this slide, we have some additional types of protozoan organisms. There's so many of them. It would be impossible for me to list them all, but they all have that very interesting trait of being able to move through their environment. Sometimes, again, it's amoeboid type movement. Sometimes, like the amoeba, the movement is based on the organism sending out these sort of extensions of its membrane that allow it to sort of crawl through its environment. But other times the organisms have actual projections like flagella, which are sort of like tails coming off of a cell, and even cilia, which are much, much smaller little hair-like structures. You can just barely see them up here in this image. And the little hairs sort of beat together in rhythmic motion and allow the cell to move through its environment. Very interesting group of organisms, the protists and the protozoa. We don't have time in this course to talk about the slime molds, but if you ever have some time available and you feel like going onto YouTube and learning a little bit about the slime molds, they are a fascinating group. Slime molds are unlike any other organism I know of. They are sort of a community of cells that live together and grow together in the environment. They can grow to a very large size very quickly. Some of them are capable of movement. They are a really fascinating type of organism, and they are protists. They don't fall into any other category. They are a group to themselves. That brings us to the end of our very general introduction to the microbial world. We've taken a look at the domains, the bacteria, the archaea, and the eukarya. And we've talked about some of the features of organisms in each domain. We've talked about the differences in the outer cell walls. We've talked about the reproductive processes that they're capable of using, what kind of energy sources they use, and other characteristics that make them unique. On this slide, we're looking at a drawing of some acellular microbes. So remember, these are the particles that are not living, technically speaking, that are still microbial to us because they are an important component of the microbial world. And of course, we're talking about the viruses, and that's what these drawings are indicative of, certain types of viral particles, and also the prions, and that's what this drawing depicts, a particle referred to as a prion. I'm not going to speak about the acellular microbes right now because we will spend time in a later lecture talking in lots of detail about acellular microbes. Let's switch gears now and talk a little bit about microbes are named. All of the cellular microbes on planet Earth have a formal scientific name. In fact, all living things on planet Earth are named using the same formal scientific system. This system was developed by a man named Carl Linnaeus. Linnaeus decided that it would be possible to group organisms together, again, based on their characteristics, based on their traits, and organize them into both a genus and a species. And if you remember, when we talked about the 7 categories of taxonomy, the kingdom and the phylum and the class and so on, genus and species are the most specific parts of that system. In other words, if you start off with a big kingdom and you work all the way down, you'll get to a smaller group called the genus, and then the smallest group called the species. So Linnaeus was the one who helped build the system of rules, if you will, that assigned every living thing to a genus and a species. Human beings, for example, belong to the genus called Homo and the species called Sapiens. So we can point to any microbial organism and we can refer to it by its scientific name. Now, one of the things we need to know about this naming system, the genus and the species, is that within a species, there can be additional divisions. And we refer to these sometimes as subspecies, but more often, in my experience, as strains. So, for example, you've all probably heard of the bacterium called E. coli. The Escherichia is the genus name, and coli is the species name for that particular bacteria. And of course, we shorthand that name as E. coli. Within that species, there are actually different variations of bacteria. They're all E. coli. They all are similar enough in their characteristics that we put them in the same genus and the same species. But there are subgroups within that species. And you may have heard of one of them, referred to as E. coli 0157H7. It's a long name, but that's the kind of E. coli that makes the news every once in a while because it's a pathogen and it causes a terrible disease, particularly in young children, called hemolytic uremic syndrome. Every year it seems like there's a report of an outbreak of E. coli 0157H7 causing disease in some local group of children. And it's terrible. These kids go through a terrible ordeal trying to recover from this illness. So the 0157H7 is the name of the strain or the subspecies of E. coli that causes that disease. When we look at particularly bacteria, it's very common that an organism has both a genus, a species, and a strain name. So something that we need to keep aware of as we move forward this summer, learning about the microbes. Now, it's important when we're communicating using writing that we write scientific names correctly. And that information is here on the bottom part of this slide. We always write out scientific names either underlining the name or putting the name in italics. The genus part of the name is always capitalized, and the species part of the name is always a lowercase. And as I said a minute ago when I was talking about E. coli, we sometimes shorthand the genus name to one letter followed by a period. So we shorten that genus name sometime for convenience sake. So when we see the name E. coli, that E stands for something. It stands for the genus Escherichia. Notice that the E is capitalized because genus names are capitalized. Notice that the species is in lowercase because we don't capitalize that. And notice that the whole name is in italics. You may have also heard of an organism called S. aureus. The S stands for Staphylococcus. That's the genus name. And aureus is the species name. You may have heard of the organism that causes tetanus, and that organism is Clostridium tetani. So in this, the way I've written it here, I've used just the initial C, and it's a capital C to represent Clostridium. And then I've got lowercase letters for the species, tetani, and the whole name is in italics. Remember, though, that there are sometimes strains involved, or again, subspecies. So you may see a name like E. coli 0157H7, the one I just mentioned a few moments ago, or E. faecalis Enterococcus faecalis, and this strain happens to be MMH594. In addition to knowing how to name microbes, the other thing that we have to do as we set off on our journey in microbiology is we have to start to think about microbes in terms of their relative size. We've said that they're microscopic. We know that we're going to need to use a microscope to visualize these organisms. But we need to also develop an understanding of just how small they really are in comparison to other cellular life. On this slide, there's a scale that runs from 1 angstrom or 0.1 angstroms on one end all the way up to 1 centimeter on the other end. So even up here at the largest measurement, we're only at 1 centimeter. And in between those two extremes, we can see a variety of different types of cells, plants and animals, bacterial cells. We can also see a viral particle on our scale, and even a small molecule. Now, the other thing I want to point out is what's under the scale. These bars represent the range of sizes that can be viewed using different types of microscopes. So this bar represents what we can view using a light microscope. And this bar represents what we can view with an electron microscope. Notice that the two bars overlap each other, number one. So yes, it is possible to see some things with a light microscope that we can also see with an electron microscope. We would just see them in better detail with the electron microscope. But there are a lot of things in our natural world that we cannot see under a light microscope. We can see our bacterial microbes, yes, but we are not able to see viral particles under a light microscope. That's a really important thing to know. We can see bacteria under a light microscope, and we're going to take a look at a lot of bacteria this summer under the microscope. But we are not able to see viral particles. They are simply too small. Now let's think about the measurements, the units of measure that we're talking about with regards to these different cell types. Plant cells are actually significantly larger on average than animal cells. Plant cells are typically in the range of about 100 micrometers. If you've never seen that unit written out in shorthand, it has a very funny looking letter. It's not a U. It's a symbol that we use to represent the micrometer. So in the area of 100 micrometers for a plant cell. And in comparison, there are 1,000 micrometers in 1 millimeter. So think about that. You've all seen rulers that have millimeter divisions on them. 1 micrometer is 1/1000th of a millimeter. Plant cells, while they are relatively big compared to other cell types, are still way too small for us to see with our eyes. Animal cells in general are about 10 times smaller than plant cells, so in the order of about 10 micrometers. And then we get down to bacterial cells, which are another 10 times smaller. So again, on average, in the area of about 1 micrometer. So 1 to 10 to 100 for bacterial cells, animal cells, plant cells. Viral particles are down in the nanometer range. 1 nanometer is 1/1000th of a micrometer. These are very, very small particles. As we'll talk about when we discuss the viruses, the particle sizes really vary. So in between 10 and 100 nanometers. And then, of course, tiny molecules would be in the range of about 1 nanometer. And those, again, you would only be able to visualize with an electron microscope.